The overall goal of this procedure is to combine transcranial direct current stimulation with proton MRS measurements to investigate the effects of bilateral stimulation on primary motor cortex metabolism. This is accomplished by first carefully positioning the stimulating electrodes over the target area and securing them to the participant's scalp while the participant is outside the scanner. After determining the position of the MRS voxel using an anatomical scan of the participant brain, 4 blocks of 64 metabolite scans are run with an MRS megapress sequence. Next, with the participant still in the MRI scanner, bilateral stimulation of the primary motor cortex is performed for 20 minutes at an intensity of 1 milliampere. The final step is to run the same metabolite scans as the pre-transcranial direct current stimulation or pre-tDCS scan. Ultimately, combining transcranial direct current stimulation with magnetic resonance spectroscopy is used to show modulations in GABA and GLX concentrations associated with bilateral stimulation of the primary motor cortex.
This method can help answer key questions in the neuromodulation field, such as which neurotransmitters are affected by specific stimulation protocols? How focal are the effects of stimulation? Does bilateral stimulation result in larger effects than unilateral stimulation? And how long are the effects of tDCS?
The implication